How can we improve photosynthesis in plants and trees with genetic engineering? Improving photosynthesis in plants and trees through genetic engineering is a promising approach to enhance crop yields, increase biomass production, and mitigate climate change by sequestering more carbon dioxide. Here are the key strategies and steps to achieve this. 1. Enhancing the Calvin cycle overexpressing rubus-CO. Background: rubus-CO is the enzyme that catalyzes the first step of the Calvin cycle, fixing CO2 into organic molecules. However, it is slow and can also react with oxygen, leading to photorespiration, which is wasteful. Approach: to overexpress rubus-CO. Genetic engineers can insert multiple copies of the rubus-CO gene into the plant genome, ensuring that the plant produces more of this enzyme. Challenges: simply increasing the amount of rubus-CO might not be enough if other factors, such as rubus-CO activase, are limiting. Additionally, higher levels of rubus-CO could require more nitrogen, potentially creating a nutrient limitation. Rubus-CO activase function: rubus-CO activase helps maintain rubus-CO in its active form by removing inhibitors. Genetic engineering: by increasing the expression of rubus-CO activase genes, we can ensure that rubus-CO remains active under various environmental conditions, particularly stress conditions like high temperatures. Challenges: balancing the levels of rubus-CO and rubus-CO activase is critical to avoid metabolic imbalances. 2. Reducing photorespiration. Introducing synthetic bypass pathways. Photorespiration: when rubus-CO reacts with O2 instead of CO2, it produces 2-phosphoglycolate, which is recycled in a process that releases CO2 and consumes energy. Synthetic pathways: scientists have designed synthetic pathways that can metabolize glycolate, a byproduct of photorespiration, into useful compounds without releasing CO2. For example, introducing genes from bacteria that convert glycolate to glycerate can reduce the energy loss from photorespiration. Challenges: integrating synthetic pathways into plant metabolism without causing unintended side effects is complex and requires careful balancing of metabolic fluxes. Photorespiration bypass. Alternative pathways: by introducing alternative pathways from other organisms, plants can process glycolate more efficiently. For instance, pathways from cyanobacteria and algae can be used. Example: the photorespiratory bypass strategy involves introducing a series of genes that reroute glycolate metabolism to bypass the traditional photorespiratory pathway, thereby reducing CO2 release and energy consumption. Challenges: ensuring that these foreign pathways are correctly regulated and do not interfere with other metabolic processes is crucial. 3. Optimizing light utilization. Broadening light spectrum utilization. Photosynthetic pigments. Plants primarily use chlorophyll and B, which absorb light in the blue and red regions of the spectrum. Expanding spectrum: by incorporating pigments such as chlorophyll D or F, which can absorb far-red light, plants can use a broader range of the light spectrum. Genetic engineering: this involves transferring genes responsible for these pigments from cyanobacteria or red algae into plants. Challenges: ensuring these pigments are properly integrated into the plant's photosystems without disrupting existing light harvesting mechanisms is complex. Improving light harvesting complexes. Light harvesting complexes, LHCs. These are groups of proteins and pigments that capture light energy and transfer it to the reaction center. Optimization: genetic modifications can be made to improve the arrangement and composition of LHCs to reduce energy losses and increase efficiency. Example: modifying the antenna size and composition to ensure optimal energy transfer efficiency to the reaction center. Challenges: fine-tuning these complexes to avoid unwanted energy dissipation or damage due to excess light absorption is necessary. 4. Increasing CO2 concentration around rubus-CO. Engineering CO2 concentrating mechanisms, CCMs. Natural CCMs. Some algae and cyanobacteria have mechanisms that concentrate CO2 around rubus-CO, enhancing its efficiency. Introduction to plants: this involves introducing genes encoding components of CCMs, such as bicarbonate transporters and carboxysomes, into higher plants. Carboxysomes: these are protein-based microcompartments that encapsulate rubus-CO and other enzymes to maintain a high CO2 concentration. Challenges: adapting these mechanisms to work efficiently in the cellular environment of higher plants, and ensuring proper regulation and integration with plant metabolism. 5. Enhancing electron transport chain efficiency. Optimizing electron flow. Electron transport chain, the series of complexes that transfer electrons during the light reactions of photosynthesis, generating ATP and NADPH. Engineering improvements, introducing alternative electron carriers or optimizing existing ones to enhance electron transport efficiency. ROS management, engineering plans to better manage reactive oxygen species, ROS, which are byproducts of electron transport and can cause cellular damage. Example, increasing the expression of antioxidant enzymes to mitigate ROS effects. Challenges, maintaining a balance between enhanced electron flow and ROS production to avoid oxidative stress. 6. Boosting sucrose and starch synthesis. Increasing sink capacity. Photosynthetic products, sucrose and starch are the primary storage forms of the carbon fixed during photosynthesis. Genetic modifications, increasing the expression of genes involved in sucrose and starch synthesis, can enhance the plant's ability to store and transport these compounds. Example, overexpressing genes like ADP glucospirphosphorylase, AGPOS, for starch synthesis and sucrose phosphate synthase, SBS for sucrose synthesis. Challenges, ensuring that increased production of these compounds does not lead to imbalances or depletion of other essential nutrients. 7. Genetic modifications for stress tolerance. Improving tolerance of environmental stresses. Stress impact. Environmental stresses like drought, heat, and salinity can significantly reduce photosynthetic efficiency. Genetic engineering, introducing genes that enhance stress tolerance can help plants maintain high photosynthetic rates under adverse conditions. Example, introducing genes encoding heat shock proteins, osmoprotectants, or ion transporters to help plants cope with high temperatures, osmotic stress, or salinity. Challenges, engineering stress tolerance while ensuring that the plant's growth and productivity are not adversely affected under non-stress conditions. Example projects and research realizing increased photosynthetic efficiency. RIPE project. Objective. The RIPE project aims to improve photosynthesis and staple crops by implementing various genetic engineering strategies. Funding, supported by the Bill and Melinda Gates Foundation, it focuses on enhancing photosynthesis to increase crop yields and ensure food security. Engineering plants. Success story. Researchers have successfully increased the photosynthetic efficiency of plants by introducing synthetic pathways to reduce photorespiration, leading to significant biomass gains. C4 project. Goal. This project aims to introduce the C4 photosynthetic pathway into rice, which normally uses the less efficient C3 pathway. C4 plants have a mechanism that concentrates CO2 at the site of rubus-CO, making photosynthesis more efficient. Challenges, transferring the complex C4 pathway, which involves multiple biochemical and anatomical changes, into a C3 plant like rice is a significant challenge but could greatly increase yields if successful. Conclusion: improving photosynthesis through genetic engineering involves a multifaceted approach, targeting various aspects of the photosynthetic process. By enhancing the Calvin cycle, reducing photorespiration, optimizing light utilization, increasing CO2 concentration around rubus-CO, boosting electron transport efficiency, and improving stress tolerance, we can significantly increase photosynthetic efficiency. These advances hold great potential for increasing agricultural productivity and contributing to carbon sequestration efforts, addressing both food security and climate change challenges. Each strategy requires careful consideration of potential trade-offs and unintended consequences, as well as extensive research and testing to ensure practical and sustainable implementation.